In this picture, the yellow color is showing the primary lymphoid organ. In my lecture of Introduction to Immunology, I have mentioned what is primary lymphoid organ and secondary lymphoid organ. Here, the bone marrow and the thymus are two primary lymphoid organs. Lymph node, spleen, a special tissue called mucosa associated lymphoid tissue like respiratory tract, gastrointestinal tract, urogenitary tract, and Peyer's patch in small intestines are under the secondary lymphoid organ. So one of the primary lymphoid organ is the bone marrow. In the bone marrow, a pluripotent stem cell called hematopoietic stem cell. From that hematopoietic stem cell, all the blood cells develop. A stem cell can self-renew and differentiate. And for the self-renewal and differentiation, a very delicate microenvironment called stem cell niche is very important, and this microenvironment or stem cell niche can be produced by stromal cells. In the bone marrow, hematopoietic stem cell produce all blood cells, but the final development of T cell occur in the thymus. So this is thymus. Thymus is present on the heart, and this is the cross-sectional view of thymus. Here you can see two important regions in the thymus called cortex and medulla. As I have mentioned that the final development of T cells occur in the thymus, so you can see a precursor cell, T cell precursor called double negative precursor T cell enter into the thymus from cortical medullary junction called CMJ. So the precursor of T cell called double negative means this cell do not express either CD4 and CD8. So these cells go into the subcapsular cortex. Here it proliferate, and after double negative the cell become double positive. It means this cell starts to express both CD4 and CD8. This occurs in the cortex region, and then the double positive cell becomes single positive, means a cell or thymocyte, actually immature T cell called thymocyte, can be either express CD4 or CD8, never express both CD4 and CD8 together. The single positive cell go through a selection process in the medulla. In my another class, I will explain T cell development in more details. So in the thymus there are three regions like subcapsular cortex, and medulla. In the subcapsular cortex, double negative cell proliferate. In the cortex, double positive cell are negatively and positively selected, and in the medulla, single positive cell go through a negative selection process. In the thymus, T cells are trained in such a way that they do not recognize self-antigen MHC molecule with high affinity. Those cells recognize self-antigen with high affinity. They go through a process called apoptosis. Only those T cells that recognize self-antigen and MHC complex with intermediate affinity get the positive signal and survive. Now, the lymph gland represent secondary lymphoid organ. Primary lymphoid organ are the generative site where immune cells develop. Lymph gland represents secondary lymphoid organ. Lymph gland are present all over our body. A lymph gland contains afferent lymphatics by which antigen and APC can enter. Contains efferent lymphatics by which the lymphocyte can go out. The naive lymphocyte like naive B cell and T cell enter into the lymph gland by blood vessels. And there are three important regions. One is called outer cortex where follicles are present. These are the follicles. Here B cell are present called B cell zone. So follicle are called B cell zone because here the B cells are present. This region is called paracortex region. This region is filled with T cells. So it is called T cell zone, and this is the medulla. So like the thymus, this lymph gland also contain cortex and medulla, but the outer cortex contain follicle for the B cell and a paracortex region for T cell. So there is an interesting fact, both B cell and T cell can enter to this artery, but they position in different location. B cell go to the follicle and T cell go to the paracortex. What is the reason behind this segregation? The answer is chemokines. In the follicle, there is certain type of chemokines that attract B cell, and in the paracortex, there is certain type of chemokines that attract the T cells. But the B cell and T cell should remain very close to each other, because for the proper activation of B cells require antigen recognition with the B cell receptor and direct contact with the T cell. So the follicle and the paracortex are very close to each other. In the lymph gland, this follicle also contain a special region called germinal center. I want to mention a follicle can be primary or secondary depending on the presence of plasma cell. As I have mentioned in the previous slide that the follicle is populated by B cells. So when a B cell present in the follicle but there is no plasma cells called primary follicle. When a B cell activated in the follicle, they produce plasma cells. This plasma cell can come out from the efferent lymphatic duct, but some plasma cell re-enter into the lymph gland and populate the primary follicle and form a special region called germinal center. So primary follicle contain no plasma cell and secondary follicle contain plasma cells, so they also form germinal center. The germinal center, affinity maturation occur. Affinity maturation of antibody means its variable region that can bind with the antibody increases its affinity towards the same antigen. I will explain what is affinity maturation in more details in my another class. The next important term is fibroblast reticular cell conduit system or FRCC in the lymph gland. Here you can see a cell called fibroblast reticular cell form a channel-like structure in the lymph node. So a lymph node is very complex in nature. Here an important cell are present in the lymph gland called follicular dendritic cell. Don't confuse with the name dendritic cell, because this follicular dendritic cell called FDC is not dendritic cell, not generated from bone marrow also. So it is a different cell, but the shape of the cell is similar to the dendritic cell, so it is called follicular dendritic cell. This cell is present in the follicle of lymph and it helps in the differentiation of B cells. The next secondary organ is spleen. This spleen contains two important regions called white pulp and red pulp. The red pulp is the site where damaged or old RBC are destroyed, so it is called red pulp. And in the white pulp, that white pulp contains two regions called marginal zone and PALS. There is also follicle present. This follicle populated by B cells, and there is a region called PALS, periarterial lymphocyte, where T cells are present. So this is B cell rich zone and PLS is T cell rich zone, and in between the white pulp and red pulp there is marginal zone. This is marginal zone. The main function of spleen is to mount immunity against blood-borne pathogen. So if there is any pathogen in the blood, the pathogen came into the spleen, and spleen have B cell and T cells and also macrophage and dendritic cell that can represent the pathogen to the B cell and T cell. There are more recirculating lymphocytes pass daily through the spleen than through all the lymph nodes combined. In our body there are huge number of lymph nodes, but the circulating or recirculating lymphocytes that pass through the spleen daily is far greater than the all combined lymph nodes migration of cells. B cell and T cell enter and pathogen also enter through splenic artery, and they first interact with macrophage or dendritic cell in the marginal zone in this area. In the marginal zone there is dendritic cell that can trap antigen and also a type of B cell called marginal zone B cell. There are three types of B cells called follicular B cells that develop in the follicle, marginal zone B cells that present in the marginal zone of spleen, and another B cell called B1 B cell. Then go to the PALS. T cell go to the PALS, periarterial lymphocyte, and B cell go to the follicle. Here is another important secondary lymphoid system called MALT, mucosa associated lymphoid tissue. This tissue also contained follicles. In this case, Pierce patch is a member of MALT. Pierce patch present in the small intestine. There an important cell are present called M cell. They lack microvilli. So you can see in the Pierce patch there is also germinal center, means B cell can populate this area. And in this case I want to just mention that you have to remember one cell that is very important called M cell that lack microvilli. All the cells contain microvilli, but this M cell do not have microvilli. Why? Because there is a special function of this M cell. Now you can see Pierce patches are covered by an epithelial layer containing specialized cells called M cells, which have characteristic membrane ruffles. So these cells do not have microvilli but have ruffles so that antigen can come into the cell. In this case, as the Pierce patch or MALT is actually mucosa associated, B cell produce one isotype, that is IgA.